Hello everyone, my name is Mario. I'm a second year student in the chemistry department, and today I'll be telling you about my research, which is putting enzymes to work using chemical biology to combat plastic waste. So, everyone has probably seen the heartbreaking images of marine wildlife entangled in man-made garbage, or perhaps of the heaping piles of single-use water bottles and other waste accumulating in the water systems. Plastic pollution has become a worldwide environmental crisis, with over 200 million tons of plastic waste currently in the oceans, and this number only continues to increase. To make matters worse, over time, this waste releases chemicals harmful to human health. Since it became a mainstream material in the 1950s, only 2% of the plastic ever produced is still in use today. The remaining 98%, the vast majority of it has been discarded, either in landfills or in the environment. While current recycling processes help remediate some of this waste, they are either environmentally unfriendly or too expensive to implement on a global scale. A better method for plastic recycling needs to be developed, and today I will share with you how my research is helping to achieve this goal. First, it's important that we understand a little bit about the chemistry behind plastics. Plastics are part of a group of material called polymers. This means that, on the molecular level, they consist of repeating units tightly packed together into sheets, like how a book consists of sheets of paper packed together. In order to recycle the plastic, these sheets have to first be separated from each other and then further broken down into individual molecules that can be reused. This is where my research comes in. I look to use the tools nature has developed and utilize them in unnatural ways to break down man-made plastic. I work with molecules called enzymes, shown as the green Pac-Man on the slide, which are microscopic machines found in all organisms. A specific type of enzyme has been discovered that can actually break down man-made plastic, like how a pair of scissors can cut through a piece of paper. However, this enzyme encounters the same problem with plastic that scissors do with paper. They can't be used to cut through an entire book all at once. This is a problem I am seeking to overcome by developing ways to separate the tightly packed plastic sheets. To do this, I submerge the plastic into an unconventional man-made solvent shown as the yellow pentagons. This solvent is able to separate the plastic sheets and make them more accessible for the enzyme to cut them up. An important aspect of this process is that both the enzyme and the solvent remain intact and can be reused for multiple cycles of the process. We wouldn't want a process designed to reduce the amount of waste and to producing more waste. Like with any research, my project has a lot of large challenges to overcome. There are hundreds of potential separating solvents in existence, and determining the right one requires a lot of testing and analysis. Additionally, I will have to engineer the enzyme so that it can do a better job of cutting up the plastic in this unnatural solvent. My hope is that by combining a natural enzyme with a synthetic solvent, I can create an efficient and eco-friendly method for plastic waste recycling. I envision that one day, instead of discarding plastic, it can be sent to recycling facilities utilizing my method to easily and quickly break it down into a reusable form. Thank you.